Okay, guys. We're back. Hello.
Hello.
Who are you?
I am Mr. Auerbach. I will be your science teacher today. Today we're going to learn how proteins get their shape, and we are going to use these to simulate proteins. Notice how they're very flexible and they can make a variety of different shapes. That is exactly what we're going to do. You have one other thing in front of you, which is this bag of amino acids. That's what these are. The clips are to stick the amino acids to. So the metal clips, these have little magnets on them like so, and so they go on like this, but do not do that yet. What I need you to do is I need you to notice that you also have, and if everybody could get one of these in front of themselves, there should be enough of those for everybody to have one at each table.
Mine's laminated.
Yes, yours is laminated. Yours special.
I'm special.
And you'll notice that on one side you have, again, the same sort of diagram with basic amino acids. And then what we have here is the shape of all the different side chains, or R groups, that are possible here. And then on this side you get them broken down in a little bit more detail. What you will also notice is that some of them have a yellow background on this, which is represented by the yellow foam. Some of them have yellow foam backing, and then some of them have a blue background, which is represented by the blue foam backing. Some of them red, some of them white.
And one green.
And actually you should have two that are green, hopefully.
Uh-oh.
Mine doesn't have a magnet.
I know. We'll work that out. We'll fix that. Anyway, what I want you all to do is, well, first of all, the white ones. I don't even know what to say. I don't even know what to say to you right now. The white ones represent the polar amino acids. The yellow ones represent the non-polar amino acids. The blue ones represent the ones that are bases, and the red ones represent the ones that are acids. And then these green ones represent a specific amino acid called cysteine, which is very important in our protein shape. So that gets its own little category. What I want you to do, you'll also notice that you have a magnetic board here, a little magnetic circle. And what I want you to do is just to get organized, I want you to take your amino acids and I want you to organize them by type. So please put the yellow backed ones on the yellow part of the circle. Please put the blue ones, the bases on the blue spot, the polar ones on the white part, the acid ones on the red part, and the cysteines right in the center where it says CYS, right in the very center. That's where the cysteines go.
Come on, the magnets are like falling apart as I'm using it.
Oh, be careful! Try not to let that happen.
Yeah. Try not to.
Okay. I don't. I'm gonna have to get some hot glue to fix those, but we'll make it work one way or the other. I have binder clips and paper clips for the ones that won't stay.
It's not two without metal.
Then just put them aside. You won't need them.
So white ones from the white background.
Carefully put them aside.
White ones from the white background.
That is correct.
I didn't do anything.
Well, okay, now you don't have to match them. You don't have to match them exactly in the sense that you don't have to match the correct one exactly to the right place. That would take us a long time, and that's just too much busy work. So just put the yellow ones with the yellow, the white ones with the white. Very nice, like so. Yes.
I don't have this today.
Okay, so what I want you to do now, has everybody done that?
Almost.
Almost.
It's not.
Are we there yet? 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 Thank you.
Oh my god.
I'm fun on vacation.
I found a magnet.
Why are you like
I've heard if I had a dose of Ritalin for every time somebody asked me that question. I was a Ritalin kid when I was young. I'm sure nobody is surprised by this.
They thought my brother had ADHD, so they put him on Ritalin, and then he like freaked out, and
Yeah.
like there was stuff like going wrong, and they were like, "Oh, you don't have ADHD."
Yeah, that's not good. Okay, let's get back. Oh, yeah. Ew. Anyway, I'm not looking at that anymore. Anyway, no, don't just stop. Don't look over there. Here's what I want you all to do.
Mr. Arbach, we're not doing that.
Okay, I'll believe you. I totally believe you. What I want you to do now is I want you to randomly arrange your amino acids, mixing them up. Don't put all the yellows in a row and all the whites in a row. Put them randomly arranged anywhere you want, any way that you want. And make sure that you use both of your cysteines if you have to. You should have to if you
can't, because one of them
I already said that, Cassie.
Well, here. Try to attach it with that.
Oh, good.
Oh, you've got one. You've got a binder clip there.
Anywhere? All of them?
What?
Anywhere?
Yeah, why not? Why not all of them?
There's
Sure.
not enough.
Anywhere. Well, put as many as you have. Well, no, don't double up. If you run out of little metal clips, then you are done with the game. But don't worry, there's more of the game to have.
Oh, I like this. This is cool.
Yes. Does that count?
Neato.
I think I got that. Yes.
What's it say?
Lovely.
You
I'm not
go
gonna hate
there. Go
You've got mad protein skills.
here.
Yeah, I do.
You might consider being a ribosome later in life.
That is what I am talking about.
That is what I'm talking about.
Biology.
No,
Biology humor, yeah. It's not very funny.
Now,
Okay.
May I have another clip?
Another one? Oh, I forgot to do that with mine.
You have to share.
Here. I have mine rigged like this. I didn't want you to do what I did because I had to put a hole in mine. If it doesn't work, you can do the same
I
thing.
got the ones on the side.
I get the clip just kind of
Okay.
on there.
Yeah, that's fine. As long as it stays
Kind
there. I'm going to replace this one. What's that?
I was supposed to replace
Here.
Nope. As long as you, I realized I didn't have either of my green ones on there. My cysteines, which I need to have.
I'm just gonna take
Mr. Arbach.
3 clips.
Nothing.
I need it.
It does not involve you, don't worry.
I'm sure.
And literally.
Pay no attention to the people at table 2.
We're on table 6.
Okay.
We're not too.
Are we there yet?
No.
No, we're
These are very annoying.
Mr. Arbach. Do you like
That's accurate. His hair is made of protein.
You're just trying to get on camera, so there we go.
Mustache.
Okay.
Oh god.
Oh no, these ones have two. Oh no, it's just
Okay, well don't worry about it too much. If you can't get it to stay on, you could also try one of those. Yeah, don't worry about it too much.
I get it.
Slayton.
I got it.
Okay, so now what we're going to learn is the 3 ways well, there are more than 3 ways, but 3 ways that proteins get their shape. And the first way is called the Sistine Bridge. Cysteine. E-I-N-E? E-I-N-E.
Got them all.
Brrrr.
Oh, so heavy.
What's
the?
There's like an echo in here, but it's sarcastic. It echoes were sarcastic.
Mr. Arbach, I can't see.
Okay, Sistine Bridge.
Sistine Bridge.
make a better door than
Oh my
we
god,
Oh.
it's not funny anymore.
Okay, so I'm going to draw something. You're going to have to draw it twice, so leave room.
Why?
Because I'm going to draw something, then I'm going to change it.
Told us to.
So cysteine, its R group has, among other things, an S and an H, a sulfur and a hydrogen. And what happens is, if you have 2 of these, like you do, a very particular thing happens. And I need to get these to show you. Much like I have here. So here's a sulfur and a hydrogen. And another sulfur and another hydrogen. Right? So basically this.
What's left with this?
There. So, right, yellow is sulfur, the white is hydrogen. And so here's what happens. is that the hydrogens leave. The hydrogens go.
Bail out.
Yeah, so this will be, well, you can sort of, if you want to, you can circle this and say the hydrogens go, or you can just write that next to it. But when the hydrogens leave, which they most certainly do, let's think about the intermediate before we draw something else. Before we draw the next part, you might want to draw this 3 times so you can show the intermediate stage. I'm just saying.
Oh my lord.
You do what you want. So now what do we got here? We've got this intermediate stage where both of my hydrogens are gone. I'm leaving. Bye. Fine.
They just bailed.
They bailed. So now I have 2 sulfurs and they're lonely. They're not satisfied anymore. They're not happy. They're sad because they lost one of their bonds. And we all know how that could be. And so what do you think happens next?
Wow.
What happened?
Wait for
What?
the...
They connect. That's right. So the sulfur's like, hey, wait a minute. You're missing a bond too. So am I. Oh, I'm so lonely. Let's be together. And so they do. And so they connect. And this forms what's called a Sistine Bridge. And this is one of the major things that gives a lot of proteins their structure, because let's do that. Let's do that. These little clips here that you have are to connect different parts of the protein to each other. Let me put my cysteine back on because it seemed to have fallen off. But anyway, so if I bring these 2 green cysteines together, which you will all also do, I put the clips, as you notice, fit handily around the tube, or whatever you call it, the protein chain, and it stays like this. And so this is one of our first levels of protein structure, is the Sistine Bridge. This is one of the major things, like in your hair, actually. The curlier your hair, the more right. When you get a perm, or when you get your hair straightened, what you're really doing is breaking these bridges. So the chemicals that they put on your hair to straighten hair opens up these bridges, and then you reform them in a different way. I don't know what I know that the process of putting the chemicals on breaks these bridges. I've never had a perm. I don't know what happens after that. But anyway. It is? Okay.
Oh my god.
Well, it is really?
It's flashing.
That's
Is
the
it?
recording, like, yes.
Hold on, I have to look.
I hope nobody's a vampire in here, because that's,
Yeah, no, there's 84 minutes of battery left. We're good. Thank you for the warning, though. I appreciate that. If it was about to go, I would have plugged it in. So, okay, so that's one level, the Sistine Bridge, where the sulfurs connect to each other, and that creates one level of protein structure. The next one is acid-base interactions.
You missed a lot. It's not my fault.
That's okay. I'm sure all the OCD people at home thank you.
That's my fault. That's a real.
Okay.
What's a real thing?
What?
It's a real, you say that a lot, but OCD is...
I know. When people say, oh, I'm so OCD, usually they're just being like, they're saying, I'm very particular about things. I know. Real OCD is a real thing, and it's very disruptive in people's lives. I know it is. You should see me walk down the hall, like, trying not to step on the cracks. Oh my god. But,
Your grandmother's
I
bed.
hate that. Holy.
Anyway.
What?
Number 2 is acid-base.
Those are 2 different things.
Yes, but instead of in proteins, instead of the acid, right, H plus, and base, yeah, but the bases are a little different. They don't have an OH, they have an NH2. But instead of the acid and the base just neutralizing each other, they'll actually bond with each other. And so this is another level at which they'll form sort of a connection.
So you want to
And so I want, what's that?
draw that?
Yeah, that's fine. Acids and base can bond with each other. And so now, Oh, mine, look at mine. Mine happens to be lined up just so, right? Your red and your blue ones are the acids and bases. But whatever you have there, you twist it around, and this is the thing. Any way you have it, just twist it so that now the red and the blue are across from each other, and you can add another clip, another bond. Mine is kind of boring because it's basically in the same shape that it was in.
Mine's basically a giant like thing that just goes squiggly. That's not particularly interesting. Are you supposed to have 2 reds or blues? I just have 2 blues and a red. And just
It's okay.
Oh, your health over like red.
Wow, everybody's is very similar this time.
And their ears.
Huh.
That's weird. Everybody's, you should have seen, this morning we did this and everybody by now had a completely different shape. That is weird. Everybody's is very similar. Yours is a little twistier. That's good. Okay.
This looks just like
It's like a
mine.
fiddle.
I understand these things.
Yours looks
You're better at science than me,
exactly
you know.
like mine, except
Anyway.
dumb.
Yeah.
That's what I was like.
They bond. It's just when the protein folds up, it's just one of the bonds that happens and it holds it together. There's not a specific reaction that you have to learn
Can
for
this
why
base
that
also
happens.
react with this one?
I don't think that would happen like that.
Okay.
I don't really know, but I don't think so.
So
okay.
probably not. Okay.
So that's the second thing. Let's move on to the third thing because our viewers at home, I'm sure, are getting a little antsy and they want to know how this ends. And not to mention that they probably just want it to end very badly, even more than you all. Although they're sitting in the comfort of their home watching this on the computer.
They're not wearing pants.
What?
Nothing. Dun-dun-dun.
Oh, I'm wearing pants. Okay. Anyway, oh god, I'm gonna get fired for this. Put some damn pants on. Okay.
Watch English for the children out there.
Okay.
Three.
So three has to do with the fact that your body's doing this right now. Your body's making proteins. When you do this in your cells, what are we made of mostly? Water.
And carbon.
Our cells are filled with water, and so when these things come out, they're going to respond as if they were in water because they're in water. And so what's going to happen? Well, you've got your polar amino acids and your non-polar amino acids, and I want you to think about what that might do for a second while I write.
So the
Interactions.
So the hydro
Stir.
attack the water and attach to it as close to it as physically can, and hydrophobic will be as far as physically possible.
That is basically exactly correct.
Yes.
What he said. So
Hydrophobic means scared because phobic.
right, hydrophobic amino acids are the yellow ones, and they're going to tend. Okay, now. They're going to tend to find a center and dive into it, and so all of the yellow ones you might picture trying to get away from the water, whereas the ones that are polar, they might pop up and try to interact more with the water. And so, like what I've done is I sort of made a dense, chewy center here that I've made my all of the yellow ones kind of dive into the center, and then maybe the polar ones might wrap around so that they're exposed. The polar ones are the white ones, and so the white ones, don't forget, they're in water, and so the white ones are going to want to be exposed to the water. And the yellow ones are kind of going to want to not be exposed to the water. There's no wrong way to do this, well, except to have the yellow ones outside and the white ones on the inside. So why don't you all? What's that?
Y'all.
The blue ones, acids and bases, they are they're charged, and but let's not worry about those so much. Like let's not twist ourselves in knots here. Anyway, thank you. I wish I could have captured that eye roll, that barely perceptible, actually not very imperceptible eye roll. But anyway, so now are we all doing that? So okay, what I want to do is I'm going to take do I have permission to scan by all of you? Is that no? I have a no on that. Okay, that's fine. I'll scan by.
Please no, I don't want to be.
You don't want to be on camera?
No,
Turn around.
so you can-
Okay, that's fine. So, all right. So we could. All I want to show is that they all have different shapes. And will you hold yours up for me there? Nice, nice, okay. And, okay, I'm coming over. All right.
Class creating atrocities.
All right, very nice. I won't go as far as you, I promise. No, don't worry, you're not gonna make it. I'm coming, okay. Hold on, here we go. Lens cover on and over. All right, hold yours up there, Slayton.
Bingo.
Okay, and so
Atrocity.
what we've learned here today is that proteins all have different shapes based on the amino acids that are on them. So, folks, everybody look at everybody else's and just realize that just based on the fact that you put different amino acids in different orders, you've created vastly different shapes. And theoretically,
Infinite. Theoretically.
well, also, theoretically, if we did this all again with the same starting point, we'd make the same shapes again. At least if we were cells and we were really doing this like it was our job,
Over and over and over and over.
as they say, it would work out the same way every time. And that's why protein shapes are very important. Carbohydrate shapes, fat shapes aren't that important. Protein shapes are. Like if this was insulin, for example, insulin is like a key that opens up your cells to glucose. And if the key doesn't fit the lock, it's not going to open the door. And so that is why the shape of proteins is very important. And even though this just looks like a big mess, this shape, or the actual shape of a protein, oh, I'm in the wrong place, hello. This shape is very important. All right, so I am going to write 2 questions on here that you and you need to answer as your assessment for this activity. And let's see, question 1 will go like this. What, that are possible? What's the best way to say it? Why are there so many different proteins that are possible?
Why are there so many different protein possibilities?
Protein possibilities.
Okay.
I'm going to move in just a second.
Should we write down the questions?
Yeah, why don't you write them down in your notebooks? Yeah, that's the first question. There's only 2. Please describe
You guys write these down?
yes. Please describe how proteins come to have the shapes that they do.
What are these called, the individual, like,
They do
things?
have
Amino acids.
amino acids, yes. And so that's pretty much it. You have 10 minutes left, 11 minutes left. That should be plenty of time for you to write. I'm just thinking half a notebook page, sort of,
Whoa.
Okay.
Whoa.
between these 2 questions.
Oh, okay, I was playing.
Steve.
Should we
Yeah.
write the question down? Wait, like, are we answering them in the same? Two separate.
Answer them separately. Two separate answers. I would say each one. This one can probably be just a sentence or 2. This one will probably take, probably about 5 sentences. It's my guess. That's just my guess. Okay, thank you, folks, and I'll give you the rest of the time to work on that. Thank you very much. Goodbye.